I would like to welcome everybody to the 11th annual Markesbery Symposium on Aging and Dementia. And as probably most of you know, we organized this symposium 11 years ago in honor and in memory of Bill Markesbery, who was the former director of the Sanders Brown Center on Aging before I became director in 2010. And we have over the years had wonderful participants and speakers, and we're looking forward to another great session today. Unfortunately, we can't be in person yet, so we've had to do this by Zoom again this year, but hopefully next year we'll be able to come back in person. So I just want to give a couple of housekeeping announcements first. So when you log off, when you're done here today, when you log off, you're going to get an automatic survey that pops up. When you log off, please fill out that survey. It shouldn't take you more than 2 to 5 minutes, but it really helps us to understand what you liked about today and what you didn't like about today and what you might want to hear next year, because we try to tailor the speakers and the topics to what our audience would like to hear. The second thing is that if you have questions for the panelists, so we will be having a Q&A session right after the keynote speaker, and we ask that you put your questions into the Q&A. So there's a button on the bottom of your screen that should say Q&A, and you just click that and enter in your question, and try to do this while the keynote speech is going on, because the Q&A session will be right after that, and then Dr. Wilcock will be looking at the questions and getting questions for our panelists ready. So let me just give you a brief overview of what's going to happen this morning. So we're first going to have a keynote speaker, and I will be introducing him shortly. This is Allan Levey from Emory University in Atlanta. After that, we will have the panel discussion and the Q&A, which is going to be moderated by Donna Wilcock. And Dr. Levy will join the panel after his talk. And we will also have on the panel Dr. Greg Jicha, who, of course, most of you probably know is our Sanders Brown neurologist and the ADRC, head of the ADRC clinical core. We'll also have Dr. Erin Abner, who is the co-director of the data management core of our ADRC and a wonderful epidemiologist. And then we also have Marie Smart, who many of you probably remember from when she used to work in Sanders Brown in our ADRC. She's a great social worker who now works at Breckenridge Christian Care Communities, and she has agreed to come back for today to be on the panel. So that's going to be fun. And then after the Q&A, we're going to have a really interesting session on the new medication that the FDA recently gave emergency approval for, which is Educanumab, or the brand name now is Aduhelm, very interesting name. And as you all probably know, there's been a lot of controversy about this new treatment for Alzheimer's. And so we thought it would be interesting and useful for the community to be able to have a discussion about this. And this discussion panel will consist of Dr. Jicha and Dr. Abner. And then basically I'll close it out at the end of that. So that's what's going to happen today, and we look forward to a wonderful session. And it gives me great pleasure now to introduce our keynote speaker, my good colleague and friend, Dr. Allan Levey from Emory. Now, I want to give you a little background on Allan, because he's an amazing scientist. He is professor and chair in the Department of Neurology at Emory. He's also the director of the Emory ADRC, the Alzheimer's Disease Research Center, which has been renamed the Goizueta, that's how you say it, Goizueta Alzheimer's Disease Research Center, after a major donor foundation gave a lot of money for their ADRC, which is great. So Dr. Levy got his BS from the University of Michigan, and then he received his MD, Ph.D. from University of Chicago. Then he did residency in neurology at Johns Hopkins, and a short stint at the NIH for molecular biology, and then he joined Johns Hopkins again as faculty in the departments of neurology and pathology. He went to Emory in 1991, and he's held a lot of different positions. He's both a wonderful researcher as well as a great educator. He was director of graduate studies for the Neuroscience Ph.D. program. He was the director of the Emory MD Ph.D. training program, and founding director of the Emory Center for Neurodegenerative Disease, director of the graduate studies. He's also the head of the Emory Healthy Aging Study, which is a huge study to follow individuals as they age. He also leads a lot of other major initiatives, such as the Accelerating Medicines Partnership, or AMP-AD, where they are looking at characterizing proteins in the brain and in biofluids, CSF, and plasma, to try to understand how things change in the brain as we get older. He also runs a relatively new program called the Open Drug Discovery Center for AD, which is trying to develop new potential drug candidates to go forward, because of course, as we all know, we really need effective therapies for Alzheimer's. So I'm very pleased to say that he's going to give us a wonderful talk called Accelerating Progress for Treatments for Alzheimer's Disease, ? So Allan, take it away.
Thank you so much for that incredibly generous introduction. I so miss being here virtually, not with you in person. I have so many good friends at UK, and Linda's right up there among all of them. And what I want to do today is take away that question mark at the end of this. I want you to hopefully share my enthusiasm and optimism at the end of this discussion this morning, why I'm so excited about the future for Alzheimer's disease. It's been quite a journey, and I've spent my entire career, in this area. And it's such an honor to, be participating in this Markesbery Symposium. The University of Kentucky ADRC, the Sanders Brown Aging Center, just played such a central role. And I would say Bill Markesbery was ahead of his time. And Linda and the whole team there continue to contribute so much progress nationally that have inspired me personally. And so it's, again, thank you for the opportunity. Let's see if I can get my slides to advance. I'll be in good shape. There we go. Let me just first mention a variety of disclosures, because I'm going to be mentioning just a few things. I want to give an overview, very high level overview, and touch on some of the exciting things that I see. Among the things that I'll mention will be, I'm going to have to set the stage for what Greg is going to talk about in the panel with the new medicine. Can't talk about Alzheimer's disease without at least introducing that. And I'll be mentioning also an advisor for a company called Cognito, which is a novel way to do treatment. So I'm just going to be mentioning some of these. And what I'd like to talk about, frame things, is, while I know you're all here because of your interest in Alzheimer's disease and related conditions, I'll just make sure that we're all sort of starting at the same place, including where we are with current treatments. Linda mentioned the Accelerating Medicines Partnership, which has been a major source of our inspiration and effort over the last 5 to 7 years. And it's important to me because it's helping us think about how to make sure that we can detect the disease as early as possible in the future, and then think about how we can expand the treatment pipeline. And so I'm just going to again touch on things that I'm sure are ongoing everywhere, as well as highlight some of the things that we're doing at our center, which complement in many ways all the exciting things happening at University of Kentucky. So it was over 100 years ago now that the first case, so to speak, of Alzheimer's disease was discovered, and it was given its name because Alzheimer's saw under the microscope these two hallmark pathological changes, these amyloid plaques and the neurofibrillary tangles, which are now pathologically how we confirm the presence of Alzheimer's disease pathology. And these have been, so while they've been recognized for over 100 years, so much of the field has focused on these two pathologies. And by doing that, our field has learned a lot about the natural history of Alzheimer's disease. And unfortunately, I know many of you know too well. This is a journey. We have spent a lot of time trying to recognize the first clinically evident time when symptoms began, which we term mild cognitive impairment. And for people, it's a journey typically that begins with progressive memory loss, expanding into other cognitive areas that become impacted and affect functioning. And too many of us think about this later stage of dementia when people really need a significant amount of help, assistance to live, to feed, to take care of themselves. But this is a chronic disease. I think is one of the things that's really changed the way we think about the disease. And even by the time people begin to present to our clinics with mild cognitive impairment, there's considerable injury in the brain. So there's already been shrinkage that's often evident, even by the time people become symptomatic. But most importantly, I think all of us are now very excited about this large window of opportunity with the realization that these pathologies that Alzheimer's described, we now know, as we'll talk about, these begin 20 or more years before the very first signs of memory loss that really become apparent. So it's, from one hand, it's a little bit frightening that changes. There are 50 million people in the United States walking around with pathology that's estimated in their brain of Alzheimer's disease, even though they're completely normal. But the important thing is that there's this long window of opportunity before symptoms begin. And that's why so much of the work that's ongoing now, I want to try to give a broad perspective on, I think, lots of opportunities to think about how to do things better. We often call this a silver tsunami because Alzheimer's disease really affects almost 50% of people after they get to the age of 85 or so. And because all of us are living longer, the population of 85-year-olds is growing rapidly. We're going to see doubling and tripling of the number of people with Alzheimer's disease and related conditions. And so this is a huge toll it's taking on society. But of course, the greatest toll we know is that to the individuals and the families that are affected by the trajectory. And it's getting worse, so we've got to change this. What's happening is we've seen the impact of important research across other areas of biomedicine, cancer, heart disease, and stroke. And as a result, fewer people are succumbing to death as a result of these other diseases. We've changed cancer from a disease that was just, everybody feared with no treatments to now a highly treatable condition for most people. But what's happening is people are living longer, surviving other conditions. Public health has improved, and Alzheimer's disease continues to increase. So we all know we're in a situation which inspires us to do better. We've got a lot of people affected by the disease. The costs are terrible, individually in society, and the treatments we currently have are just inadequate. And with all this background, our country as well as others have begun developing national plans. And so we're at the 10th year anniversary now of the national plan in the United States that was initiated when Obama signed it into law. And the goal, the first goal of that is to prevent and effectively treat Alzheimer's disease by 2025. So already 10 years into this journey, and we have a lot of work to do if we're going to achieve this goal of preventing and effectively treating Alzheimer's disease. The Sanders Brown Center, our center, and others have been really fortunate because the increased awareness of the problem of Alzheimer's disease has led to this major growth of research dollars available. And so here in 2021, we have now over $3 billion a year going into research in this area, which is still not enough, but it continues to go up and is expected to go up because of the bipartisan support. And every couple of years, there's a convening of scientists and clinicians from around to help establish the priorities for research, to make sure that under-researched areas are invested in that have great promise. And all of us in the field have been beneficiaries of this infusion of dollars and the research opportunities that are prioritized. This is just to remind everybody that we have a lot we can do right now. When we do early diagnosis, I've always been a strong believer that that's absolutely essential. It pains me that we don't have better screening in the elderly for cognitive impairment, because there's so much that can be done. Establishing a correct diagnosis to rule out other conditions, and when Alzheimer's disease and related conditions are ruled in, we have treatments which are, not as effective as we want, but they've been around for nearly 20 years or more, including the cholinesterase inhibitors that are known by names like Aricept, et cetera, and Namenda is another class of drugs which is available. And Alzheimer's disease is, I use that term Alzheimer's disease to really reflect a broad spectrum of symptoms and pathologies that occur commonly with the memory and cognitive changes. So we know depression and sometimes delusions and hallucinations can appear as part of the disease. And I think we also don't fully utilize non-pharmacological strategies that could be helpful in many cases to help people develop tools to empower themselves to improve their memory or keep it stable, and to adapt as their memory or cognition changes. And we know many of you out there are in the role of caregivers, and there's a lot of research that needs to continue in this front as well, as we learn more about the needs that all of you have and how we can best meet them and address gaps. I always like to convey a lot of hope and encouragement. I think this is, again, a chronic illness, and there's so many years that people can do really well while we look hard and fast for better treatments that can hopefully prevent things in the future. So I like to end this treatment part by saying, we really haven't had any disease-modifying treatments that are approved. Why is that? And as I mentioned, we've known about these amyloid plaques and neurofibrillary tangles for 100 years, but the world has been focused on one idea, which has been an important idea, and it's because of the recognition that amyloid plaques are composed of this protein called amyloid, or A-beta peptide, and it's led to this whole theory that has galvanized the field and industry to think about how we can get rid of the amyloid plaques. And I think everybody has woken up after the last 20 years of clinical trials, which this is a list of some of them, which I don't expect you to read here. But every single one of a multi-pronged approach to treat these amyloid, to prevent its production, to help clear it away out of the brain, and other strategies have really all failed. And that is what inspired a lot of the strategic plan nationally, that we've got to broaden the search and understand the complexity of what's happening in the disease. And just a couple of years ago, we started seeing really concerning signs that even the major pharmaceutical companies, who we absolutely depend on to help bring drugs to the clinic, started getting concerned. Because of the failure of the clinical trials and the large cost that goes into these clinical trials, people really started getting concerned. There was alarm went up when Pfizer, one of the major drug companies, halted their research program in Alzheimer's and Parkinson's disease, and others stopped major programs. And so our whole field changed quite a bit last year in a very surprising FDA decision to consider approving this drug that Linda mentioned at the beginning called Educanumab, or Educanumab. And it's a monoclonal antibody to the amyloid, much like many of those failed treatments. But this study, you're going to talk a lot about with Dr. Jicha in the panel later. And I just wanted to set the stage for that panel because, again, it's so important. The reason people have been interested in this drug, there was a phase 2 study, a small study, which was not meant to determine whether the drug would be approved or not. But it illustrates nicely what this whole class of drugs now is pretty well established to do. And what you're looking at here on the left are brain scans from 4 individuals in the phase 2 study. And you're looking at the yellow and red, which is the amyloid signal. It's where the amyloid plaques are developing. And you can see at baseline, when these individuals started, they all had lots of the amyloid present in their brain scans. And after 1 year of treatment with this drug in this study, those who received the placebo or didn't receive the active medicine had no change or worsening on the amount of amyloid that was accumulating, but in a dose-dependent way, the amyloid was cleared. And they also showed in this phase 2 study some promising results for the first time that this change in amyloid was actually showing some relationship to the progression of dementia in a dose-dependent way. So that was very encouraging and led to the trials that Dr. Jicha is going to talk about that have become very, very controversial. And again, I have been a consultant for Biogen, so I just want to make sure that's disclosed. They probably don't like that I'm a consultant because I haven't been terribly enthusiastic. But basically, this was what they did was 2 large clinical trials where over 3,000 participants from many of the Alzheimer's disease research centers and other places around the globe participated, where they received this monoclonal antibody to amyloid for about a 1.5 year period. And one study was called Gage, and one study was called Emerge. And what was shown was what we have seen across many of these anti-amyloid trials is that the amyloid can be cleared. And I'm not going to go through the details of this, but remember there were 2 trials, and one of them showed what you're looking at here. I think is important to look at. And so the purple line going across here, those people who received the high dose of the monoclonal antibody, and the placebo here is in gray down here. And what we're looking at is over time, the mean change in the memory or cognition on a scale called the mini mental status exam. And over time, people decline, as we know happens with Alzheimer's disease. And there's this very small difference between the placebo and the treatment, which is what has been considered the benefit. So it's a very small change, and people don't often recognize that continually the disease is still getting worse. So it's not reversing anything at all. And in this one trial, there was also some small benefit seen in how people were doing functionally. They did not lose functional abilities quite as fast as those in the placebo. But the almost identical study showed 0 difference between the treatment and the placebo. So that's why there's been huge controversy, which will be discussed in the panel, because usually the FDA requires 2 trials like these to both show significant benefits. And so again, that's where we are. And as Linda said, the FDA has approved this as a sort of conditional or fast track pathway where more data needs to be accumulated. So I consider that we still do not have any disease modifying treatments at this point that are clearly effective. Our own practice at Emory, we're not even prescribing this yet. For patients, we may or may not do that. If we do so, it's going to be under only in very, very restricted conditions for people that would otherwise have qualified for this clinical trial. So you're going to talk a lot more about that. So that's pretty much all I want to talk about amyloid, because the exciting part, I think, is thinking about this going forward. But why are these trials all failing for the most part? And one of the major reasons is because the symptoms are occurring, as I showed you earlier, after a long 20+ year period when the pathology is there. And so we're starting these clinical trials very late in the process. Many of us think there are other targets that are important to go after other than amyloid, and we'll talk about that. And we still have a heck of a lot to learn about really what's causing the disease, particularly in these early stages of disease. And so part of this strategy then was to expand the pipeline of novel therapeutic approaches. And one of the initiatives that has been funded is called the Accelerating Medicine Partnership. And it's a partnership between several academic groups, as well as industry and NIH, to work together in an open science manner to produce data that anybody can access to help us learn about what's going on in the disease. And this program was inspired by thinking about the really exciting progress that's happened over the last 10 years in understanding the causal mechanisms involved in Alzheimer's disease. And we know this is a complex disease, that it is a combination of multiple genetic influences, the environment we live in, and unbiased approaches to try to find out what's going on have used genetics. So thousands of people are necessary to participate in genetic studies so we can understand which genes confer risk. This is even a short list now. There's nearly 40 different genes which we all carry that have different flavors. And depending on the flavors of those 40 genes that we have, our risk for Alzheimer's disease is either increased or decreased. So this is something we're born with. And epidemiologic studies also provide really a much more of an unbiased approach by studying large numbers of people and understanding what factors lead to people having an association with increased or decreased risk. And a whole host of things have come out repeatedly from different epidemiologic studies and different cohorts across different parts of the world. We know that age, by far, the older we get, the greater our risk. We know there are influences of gender and many other factors. And in green here, I'm highlighting that there's a lot of evidence that continues to accumulate that education, exercise, anti-inflammatory diets like the Mediterranean diet or the MIND diet are all things that can influence risk in a population base. So this is all what we consider association. So it's not necessarily cause and effect, but they're important clues. And if you look at this holistically, we've been focusing as a world on these amyloid as a target. The genetics has only pointed towards amyloid in a very, very small number of people with an extremely aggressive form of young onset Alzheimer's disease, and much less in the more common forms. But if we look at what these genes and epidemiologic studies are suggesting to us, that many of these genes and risk factors seem to be associated with lipid biology, vascular biology. Dr. Estes and others at Kentucky have done great work helping us understand how these genes confer risk of the disease. And then there are also many genes and factors in these studies that suggest that inflammation is another one. So again, this inspired the Accelerating Medicine Partnership Program, that we need to actually start with human brains. And there's nothing more informative than studying a person during life. So many of generous participants in the Alzheimer's disease research centers donate their brains at death, as well as controls. So people who have had different diseases as well as who are just healthy. That has become one of the most important sources of information for us to learn what's really going on in the brain. That we can then turn that information into better ideas to test other things in basic research studies. So we've now, as a consortium of different centers that are part of this Accelerated Medicine Partnership Program, we have actually done analyzed over 2,500 postmortem human brains, where there's a wealth of clinical information available, detailed pathology that's done consistently, genetic information, and all these other molecular fancy ways to understand at a very precise level what the changes are occurring in the brain. Our team at Emory has been leading this consortium for proteomics, and I'll try to explain what that is very briefly. This is a cartoon showing how every cell in our body has a complete replication of the 20 to 25,000 genes that make us human. Every single cell in our body has that. And we're born with that blueprint of life. And each of those cells changes that genetic information into a product, and the product is a protein. And while there are 20 to 25,000 genes, these confer the instructions for cells to make over 1 million different forms of proteins. So it's very, very complex. But it's these proteins that are the ones that are the substance, the architecture of the brain cells, the immune system, and the amyloid plaques themselves are a protein. And the inflammation that occurs around it, and many are protein mediators of these things. And so proteins really, in many ways, are one of the final common steps in the execution or pathway, as I like to say. And so these proteins reflect the integration of the genetic and environmental influences that we just talked about. This is not going to be a scientific talk, so I'm not going to get into all the details. But this approach, allowing us to study for the first time at scale, we can measure about 10 to 12,000 proteins now in these brains, and thousands of brains, and begin to understand what the changes are. This has led us just over the last couple of years to be able to have some very different insights, I think, into what's going on, which almost always has been complementing and extending what others have been studying for other approaches. For example, the proteomics that we do from analyzing brain has shown us that there are clear signs of changes in brain metabolism, as well as brain inflammation. Again, Dr. Eldik, Dr. Estes, others, Dr. Wilcock, there's so many great things that this team has been doing at UK, looking at brain inflammation and other things. And this allows us to now extend that work into identifying individual proteins, or what we say are a community of proteins, hundreds of proteins that can reflect new biomarkers or new therapeutic targets. And while this starts with postmortem human brain, as I'll show you, we've also spent a lot of time trying to translate this into tools that we can study in living people, so that we can use these for early detection of different processes that are going on in brain. And we can also extend the genetic study. So I mentioned the 40+ genes, which now show risk genes. There are a lot more. That's in many ways the tip of the iceberg. And what we can do now is you do these sophisticated analyses where we can identify which proteins are individually mediating the genetic influences on the disease. And many of these proteins we can begin to now associate with other conditions, including symptoms like depression that occur in the disease. So these are just all the sort of things that have come up from our group over the last couple of years. To just give you a highlight of one type of example, across these brains, we have seen one group of proteins that we were unaware of previously. Their individual proteins are shown here in this circle, and their identity is not so important individually. But these 100+ proteins all seem to behave together across all these thousands of brains we've looked at. And where studies are available, what we're learning is that many of these proteins reflect the immune activation, the inflammation that occurs as part of the disease process. And this is we've seen this in multiple cohorts of hundreds of individuals from across the country. And this is one study from Seattle, a longitudinal study called The Adult Changes in Thought. And what we learned was that this community of proteins increases quite a bit in Alzheimer's disease brain, shown here, versus control brain. But we've also been studying a group of brains that are from people who were studied annually during life, like many of the participants in the Alzheimer's centers. But then at autopsy, after they passed, it was found that they were cognitively normal, but their brains had Alzheimer's disease pathology. So we call this asymptomatic AD. And we think this is in part due to this preclinical stage of the disease. So this group of proteins is changing before symptoms begin. And there's a very strong correlation with the diagnosis. And then yet another cohort from one of our colleague Alzheimer's disease centers in Arizona, we see the same group of proteins as a very strong correlation with cognitive ability. And so the higher the amount of these protein levels and these signs of inflammation, the more impaired people were cognitively. So these strong associations then give us clues where the next step has been to actually do studies and basic studies, which I'm not going to go into, where we can manipulate each of these genes in animals like fruit flies and mice, or in cells that we derive from humans, to actually determine individually which of these proteins, if any, may be relevant for new treatment approaches. So this, just to summarize what we're learning from our group and from others, and really the worldwide effort from the increased expansion of knowledge about basic science, is that, while this is a complex disease of genetic, environmental, lifestyle interactions, and now social determinants of health, there are a lot of things that are contributing. And there are a lot of complex pathologies happening in the brain. It's far more than amyloid, which is early on this long trajectory that goes from healthy aging to Alzheimer's disease. Dr. Jicha and others at Kentucky have just done a magnificent job helping us learn about the changes in vascular function and blood flow that are also part of this. And much of the cognitive impairment that occurs in stroke and cerebrovascular disease is very much related. Again, the team has done a great work here at UK on inflammation. Everybody around the world is waking up to the importance of inflammation, how complicated this is. So it can be either damaging or protective. And we've got to learn the nuances to really turn that into important therapeutic approaches, as Linda has really nicely shown us. Brain metabolism, neurofibrillary tangles. And then I add this in Kentucky blue for my friend Pete Nelson, the head of the Neuropath Corps at UK. There are pathologies we didn't even realize that we still have so much more to learn. There's a protein that accumulates called TDP43, which Dr. Nelson has really helped organize globally our understanding of how this pathology also contributes to ultimately the cognitive impairment due to synaptic loss and neuron loss. So this is a complex process, and I take that as a huge opportunity for us. And because it gives us a whole new set of ways to develop tools to improve our ability to detect the disease as early as possible and to turn that into treatments, hopefully as early as possible. And if we can do that, if we can start treatments, 20 years before symptoms are beginning, we can even have sort of weekly effective treatments, but we can push things out. So if we can get people living to 90, 95 without cognitive impairment, we'll let nature take its course and really hopefully protect the golden years for all of us as we age and the next generation. So let me segue now to briefly talk about some examples here, how we can turn this early detection, turn this knowledge into ways to improve early detection. Common ways that are being developed and now used pretty widely across all the Alzheimer's disease research centers and often in clinical practice are brain imaging and spinal taps to measure spinal fluid. I know those are common at UK as well. But there are advances going on across the scope of understanding this disease. We now very well can identify those core pathologies, amyloid plaques and neurofibrillary tangles, with these sophisticated PET scans. We're not huge fans of those at Emory because they're so expensive. And I always try to keep my eye on what's going to disseminate worldwide in an affordable, scalable way for treatment. And these things are just not available in most places, and nobody's going to be able to afford them. But they're absolutely critical research tools for us because they provide really what autopsy used to provide, sort of gold standard diagnosis. Our center relies heavily on spinal fluid because the spinal fluid bathes the brain and allows us to measure proteins that are floating around in the spinal fluid, which are reflective of these two core pathologies. And as I've just mentioned, our group is very interested in understanding things beyond those two pathologies. I showed you those 100 plus proteins which are present in the brain, even in an asymptomatic stage, that seem to be strongly related to cognition. We started trying to target novel proteins that are also measurable in the spinal fluid. And we took 28 of these proteins that are present in brain and developed ways to measure them in the spinal fluid using mass spectrometry for those who are scientifically inclined. And what we can do, we've seen in early studies that this was going to be a very promising approach, that we can measure proteins and correlate what's in the spinal fluid with what's in the brain, so that these become biomarkers of the actual changes that are occurring in the brain. So over the last few years, we've been spending a lot of time trying to better understand what changes occur in proteins in the spinal fluid. And we've identified really hundreds of proteins which are changed as a result of Alzheimer's disease in the spinal fluid. And because we have a good understanding of the protein changes in brain, we're able to map them. Again, I'm not going to go into details here for the sake of time and understanding who I'm speaking to, but we can now, we sort of reduced a large amount of data so we can begin to think about different proteins that reflect different complex biologies occurring in brain. Some of these proteins reflect changes to brain synapses. Others reflect changes in the cerebrovascular function or blood-brain barrier. Others reflect the white matter. Others reflect this inflammation, and others reflect metabolism. So we're developing these precise protein targets that relate to these different functions, which I showed you are now known to be important in the complex disease pathology. And so we can measure these proteins now and show that they're all in the spinal fluid very, very significantly changed between individuals who don't have Alzheimer's disease and those who do have Alzheimer's disease. And so each of these panels of proteins shows very, very dramatic differences between disease states. One of the biggest challenges we've had in the field is to replicate things, to make sure we can get large groups of people together from across centers and confirm things. And these protein changes in the spinal fluid we've been able to show across 4 other replication studies, different cohorts now, that this is a very reproducible finding. So I'm going to, just for the sake of time, pass on that. One of the, what we wanted to do to be able to scale this is we've adapted a more targeted method, which allows us to measure individual proteins at scale pretty rapidly. And so one of the studies that we're involved in is what's called the autosomal dominant form of Alzheimer's disease, where all of our centers get together and get these rare cases where there are mutations in the amyloid precursor protein or presenilins that cause this young aggressive onset form of Alzheimer's disease. And in these studies, we know there are many young people who are not even symptomatic, but some carry the gene, which we know is going to cause them to develop Alzheimer's disease in the decades ahead. Others in participating in the study are siblings who do not have the gene. So we've been able to study almost 900 spinal fluid samples from these individuals over time and test how these new protein biomarkers in the spinal fluid that we've discovered, how they perform, how they might be able to predict Alzheimer's disease. And we're very encouraged because many of them are showing changes and telling us that the biology, like inflammation or metabolism, are actually getting changed in living people before symptoms begin. So these become new biomarkers as well as potential targets, so we can monitor whether new therapeutic approaches are working or not. The really exciting progress in the field has been, we recognize that spinal fluid is not something everybody wants to give up. We do it routinely, as you guys do at UK, but clearly we would all love to see simple blood tests, which can provide the same benefit. And the field has seen just tremendous progress. So we really are on the precipice of having available in clinic and research studies simple blood tests that will let us measure both the amyloid plaques and the neurofibrillary tangles, I think, in the years ahead. And the same pipeline that I've shown you for looking at these other biomarkers in spinal fluid, we're now beginning to do in blood as well. And we've had some pretty good initial success. So we're hopeful that we'll be able to, in the future, envision a whole panel of blood tests, which can help us understand the amount of inflammation or metabolic changes or synaptic injury that are occurring together with the amyloid or the neurofibrillary tangles that define the disease. Most importantly, we all want to be where we can actually do more than detect the disease. And the reason we do this is so we can think more intelligently about how to intervene. And it's really important to remember that we have a lot of risk factors that are identified It's a real challenge for the field to turn risk factors into treatment approaches, but it's a really important area that needs to continue to grow. As I showed you, the epidemiologic studies, there are a lot of risk factors that are identified, and there are a lot of protective factors that are identified. The way I think about these is that they sort of are in a balance and push-pull in terms of determining when somebody is going to get the disease or how rapidly it will progress for them. So these are things that are potentially modifiable. And one of the most important studies, I think, over the last couple of years, the clinical trials, was something called the SPRINT trial, which had over 8,000 individuals where blood pressure was being assessed. And there's been a lot of questions about how aggressive we should be with treating blood pressure just for medicine. They're really designed to protect the heart and protect against stroke. But these 8,000 plus individuals were followed out over years with either the standard care where blood pressure is allowed to, basically be treated to a systolic up to about 140, or more aggressive blood pressure control, where the goal is to get blood pressure systolic under 120. And this was really the first study that I'm aware of that really convincingly showed how important blood pressure control can be. Because what it showed is that there was a reduced risk of progressing to mild cognitive impairment and dementia in normal people as a result of the aggressive blood pressure control. So that's something that can be done today. So I encourage people to be aggressive about getting their blood pressure controlled. And I was really delighted to see Donna, and the team in the UK participated, excuse me, in thinking about how these blood biomarkers are going to be a new generation, new opportunity to understand who should be treated and when they should be treated, and if the treatments are effective enough. So I think these biomarkers are going to be very complementary to thinking about how lifestyle might modify risk. And so there are a whole host of things other than blood pressure which have been identified as potentially modifiable, including education we've known has a big impact. There are clues that even hearing loss in midlife could be a risk factor for Alzheimer's disease. And in later life, depression, physical inactivity, and social isolation and smoking are all really important risk factors that are all potentially modifiable. So there's a lot of work that needs to be done, but most of these things come with zero risk and a lot of general health benefits. And so I think the more we can encourage people to adopt these healthy lifestyles throughout their life, the better we're going to be. We also know that lifestyle is not going to be the end all. We need to develop better medicines, without a doubt. And this is a figure from a review from one of our colleagues, Dr. Jeff Cummings, who's led a lot of clinical trials in our field, who periodically summarizes all the different treatments ongoing. The only point I want to make from this slide is that we now have over 120 different clinical agents, drugs, therapies being tested at various stages, from early stages to late stages. I think there are at least a couple drugs developed by Dr. Van Eldik that are now in humans as well, that are on this. And so this pipeline has to grow, but I'm very encouraged to see that the numbers of new things being tested continue to increase every year. It's a long slog developing a new drug. I'm sure Dr. Van Eldik will confirm that. It's often said to be a 20 to 25 year journey from the time somebody has an idea to develop a new drug to a new protein target, for example. One of the things that I think our field is underappreciating is the opportunity to repurpose medications. In the cancer field, a lot of things are being tested that have been FDA approved for general medical conditions like blood pressure or diabetes control or antidepressants, etc. And in cancer, these things are often being now tested and sometimes being found to be effective for various forms of treatment. These exact same mechanisms provide opportunities for Alzheimer's disease that I think are underappreciated. My colleague at Emory, Dr. Ihab Hajar, is a geriatrician and has been studied cardiovascular risk factors for many years. He has really helped me open my eyes to how important these can be. And so Dr. Hajar looked at our our Alzheimer's Center participants from across the country, including at UK, and looked at those who were taking one of these blood pressure medicines. It's a certain class of medication called renin-angiotensin blockers. And so this class of medicines, what you're looking at are people in the national consortium called NAC, who were non-users in blue, and the people that took this one class of blood pressure medicines in red. And what Dr. Hajar saw is that the progression of their dementia, the severity of their dementia rating, was less for those people taking that medicine. He also used one of the other national studies that we all participate in called the ADNI. So these are, some of you may be participating in that, where you get repeated brain imaging, some people get spinal taps. And EHAB looked at this cohort and found that those people that were taking the angiotensin receptor blocker medicine class of drugs actually had less progressive increase in their tau, one of those spinal fluid measures of the neurofibrillary tangles and associated with the amyloid plaques as well. And so this was encouraging sort of association data. It inspired him to do a clinical trial that we finished at Emory. He took 176 people with hypertension and then looked at, so these are people not necessarily even with the diagnosis of Alzheimer's disease, but mild cognitive impairment due to and hypertension, and treated them with this class of medicine specific drug called candesartan. And what EHAB found in this phase 2 clinical trial is that the candesartan actually showed some improvement, which is this reduction in time it takes to do this attention task or this sort of executive function task that we call TRAILS-B. So there was a symptomatic benefit in this clinical trial as a result of treatment with this one class of medicine, candesartan, versus another blood pressure control. And also he's found some benefit in the white matter, suggesting that there may be some protective effects in the brain as well. And then I know I just have a few more minutes left. I'll just highlight another repurposing effort that we did at Emory based on a drug called Animoxetine. And so Animoxetine is an FDA-approved drug for attention deficit disorder. And it's been used safely in children and adults for many years. But it targets a very selective pathway that's been of interest over the last couple of years in the field. And this is really work that was led. We were inspired by my colleague, Dr. David Weinschenker at Emory, who has done a lot of the basic research understanding this group of brain cells down in the brainstem called the locus coeruleus. And this group of brain cells secretes a neurotransmitter called norepinephrine. And this norepinephrine bathes a lot of the brain. It connects with many parts of the cerebral cortex and the parts of the brain involved in learning and memory. But as Dr. Nelson knows, our neuropathologists have identified that this group of brain cells you can see because it has a pigment in it that allows you to visualize it. And that's what these black spots are deep in the brainstem. But in Alzheimer's disease, this group of brain cells is really severely devastated and very early. And one of the most interesting things that we've been learning as a field is this group of brain cells seems to be the first site in the entire brain that develops some pathology, the neurofibrillary tangles. Even in young adults or even youngsters begin to form some pre-neurofibrillary tangle-like changes in this group. And a whole variety of basic research studies have shown that when this group of brain cells degenerates, there are a lot of things that happen throughout the brain. It activates inflammation, it changes the blood-brain barrier, changes the blood metabolism, all the things that are somewhat similar to what actually happens in Alzheimer's disease. And then in the experimental models, it's also been shown that drugs like Animoxetine, which increase the amount of norepinephrine released by these brain cells, can actually rescue some of the symptoms as a result of degeneration of those brain cells. So as that work progressed, we realized it almost felt unconscionable that we shouldn't be trying something like this, which is already approved by the FDA. So we did a clinical trial where we identified people with mild cognitive impairment. And these were people who had their early memory loss due to early stages of Alzheimer's disease that we confirmed by spinal fluid. And we treated them with either Animoxetine or placebo, and everybody got both. So it's what we call crossover. We were able to measure that The drug got into the brain and had very expected strong effects on norepinephrine metabolism, as we expected. I was quite surprised. We actually saw a reduction in the amount of the spinal fluid measures of neurodegeneration, or the tangles, the phospho-tau and tau, in this trial. And we also did fMRI to look at brain metabolism, and FDG PET, two different ways to look at sort of brain metabolism. And what we found is that there was an effective treatment on brain metabolism as well. And then these biomarkers that I've been showing you in the spinal fluid, we found significant beneficial trends with many of these panels that I talked about as well. So this was just accepted for publication. So hopefully we'll get this out there, and we'd like to see this replicated and go to the next level, again, as a way to help accelerate and broaden the pipeline of treatments. And final, as a really cool study that we got involved with when a young faculty member moved to the biomedical engineering department at Emory and Georgia Tech a couple years ago to look at non-invasive treatments, which I think have more promise than I realized. And this is Dr. Annabelle Singer. She came after her postdoc at MIT, where she had discovered that if she could create she could stimulate brain cells at a certain frequency, so that the brain cells would fire 40 times per second, seemed to have a very beneficial effect on memory and learning, and that she could induce this electrically, but more shockingly, she could flash lights to these mice and get the brain to start firing at the same frequency as the light flickering at 40 hertz. So it really suggested that if you could stimulate the brain non-invasively with lights, or she also then showed sound, you might be able to modulate brain activity. And what was pretty impressive is they did this in these Alzheimer's disease mouse models. These mice accumulate amyloid plaques, shown here in green. And by flickering the lights or sound for these animals, they were able to show there was less amyloid that accumulated, and that the brain cells, the immune activation occurred as a result to help seemingly clear out the amyloid. So it started suggesting a possible mechanism by which flicker might be actually able to change the actual disease pathology, which is, to me, quite remarkable. So this was early work, but we decided that, again, it's very safe, and we wanted to test the potential to do this in humans. So Annabelle, who's a biomedical engineer, came to us, and we designed a clinical trial. Colleague Dr. Jim Law helped lead the design of this. This is just very simple. We identified 10 participants who wanted to get involved in this to see if this would even be feasible and tolerated. And so what we did is we hooked these people up with the glasses and earphones and had them take them home so they could use this for an hour a day. And we had a couple parallel groups of the participants where they either had them but didn't have the flicker at the right frequency, or they did at various points in the trial. And we found that people loved doing it. They stuck with it throughout the up to 10 months that people had been studied. We did some of these other similar studies. We did fMRI studies where we look at brain activity across the regions by MRI, and saw the treatment was actually improving the connection of two of the important brain regions where there's changes in Alzheimer's disease. We did the same proteomics approach, so we looked in the CSF, the spinal fluid. We could measure the proteins in the spinal fluid and classify pretty accurately whether people were during the placebo phase of the treatment or getting the stimulation. And then one of our colleagues, Levi Wood at Georgia Tech, measured a whole panel of immune markers, measures that the immune system was getting activated, and saw a whole variety of changes. So to me, this is very encouraging that even non-invasive approaches have true potential to modulate real biology, real physiology, and potentially even pathology. I think there's going to be a lot of excitement in the future thinking about a whole variety of approaches, and not just pharmacologic. So as I conclude, I hope I've been able to show you that Alzheimer's disease is a complex disease. There's a lot more going on in the brain than the amyloid plaques and the neurofibrillary tangles that we've known about. that the scientific advances are just so exciting. I can't tell you how there's never been a more exciting time to be a scientist and clinician because of all the great work going on all over. We're learning daily new things about the genetic causes and the way the genetic causes are transformed into the disease, and how we might capitalize on learning about that to identify new therapies and biomarkers that are going to help us detect the disease early, even pre-symptomatically, without a doubt, in the years ahead. And that our pipeline is going to continue to grow. So I am, for one, I'm very optimistic. And none of this happens without great researchers like those at UK and great participants like all of you. This has got to be a partnership, and it's so important that we get people to help us in this journey, because working together, we're going to get to a cure faster than ever. So with that, I just want to acknowledge just a fantastic group of people I get to work with at Emory. I won't go through their name, and the programs like the Accelerating Medicine Partnership that I talked about have colleagues from really across the country. And we also have industry partners that have put in additional money into the program other than the NIH. So with that, just thank you all for attention and giving up some of your Saturday morning, and look forward to participating in the rest of the discussion.
Great. Well, thank you very much, Allan. And I think you're right. I mean, we are accelerating progress for treatments for Alzheimer's disease. So we definitely can take that question mark off of your title. So now I'd like to turn the program over to Dr. Donna Wilcock, who is going to be leading the Q&A. She's going to be moderating the panel. And just to remind you, I mentioned before, but Donna is the head of our biomarker core for the UK ADRC, as well as the director of the MARC-VCID biomarker initiative, which is for vascular cognitive impairment. And she will be moderating this Q&A. So if anyone has questions, please put your question into the Q&A, hit the Q&A button or the chat button, and we'll try to get the panelists to address your questions. So take it away, Donna.
Thank you so much, Linda. Before we get started, I wanted to ask the panelists, just give yourselves a brief introduction, your name and what your area of expertise is, to give the attendees some idea of who everybody is. So we'll start with Marie.
Good morning. It's good to be here with you again. I appreciate the invitation. I am Marie Smart. I was for 29 years a family support person at the Sanders Brown Center on Aging with the Alzheimer's Disease Center, and still feel very much a part of that team, and appreciate you all seeing me somewhat in that sense. I am retired from UK and am still providing support services to families privately in the community, as well as consulting with some dementia-specific long-term care settings, including the Breckenridge and some of the Christian care facilities in Kentucky.
Thanks so much, Marie. And Dr. Abner?
Good morning, thank you. I am Erin Abner. I'm an associate professor of epidemiology and biostatistics. I am the co-lead of the data core of our ADRC, and I also do a lot of work in clinical trials.
Thanks so much. And Dr. Jicha.
Hi, I'm Dr. Greg Jicha. Welcome this morning. And Dr. Levy, that was just a wonderful introduction. Really something that we've been working on for years is this inspiration of hope, really letting people know how close we are coming to better medicines for disease. So I'm delighted to be here with you all today, and my colleagues, Marie, Erin, Allan, and Donna, as well as Linda, our center director.
Thanks so much, everyone. So we'll kick this off with a question for Dr. Levy, since this relates to your presentation. Someone would like to know about physical inactivity related to cognitive health in late life, and how in the studies you described, how are you defining physical inactivity? And does it mean that late life inactivity might do more harm than midlife?
Great questions. I put physical activity as one of the areas that, again, needs a lot more study. There certainly have been clues from some trials in mild cognitive impairment, for example, that introducing exercise even at that stage may be of some benefit. I think the way I think about these things, and certainly make my recommendations daily in clinic, are to follow the American Heart Association guidelines for how to define exercise. And so I define it as their goals is 150 minutes a week of exercise. And I think it's really a lifelong, the more lifelong behavior one follows, the more likely it's going to be effective. I think we're learning, but it's certainly not too late in life to increase exercise as well. And I think it's a double-edged sword. There's physical activity or physical inactivity. It depends on whether you're an optimist or a pessimist. But we've all known in medicine that people who are bedbound and stop exercising, their muscles can start wasting away within days. They can become weak really quickly. And I think that becomes dramatic, but we're less, I think it's not quite as dramatic when you increase exercise that you're going to see this proliferation, but certainly the old adage, use it or lose it, I think holds up for exercise as well.
Thank you so much for that, Allan. So I want to try and get to as many of our questions as we can. This would be a question for Dr. Abner. Do we know how alcohol use affects the progression of brain diseases like Alzheimer's?
I'd say we don't really know all that well, and it's important to remember that alcohol use isn't something that's either there or not, right? There's a large range of behaviors and doses that we could mean when we're talking about alcohol use. So you may have seen on some of Dr. Levy's slides, I think there was a reference to red wine potentially being protective. That certainly wouldn't be true if you were drinking 4 bottles of red wine a day, for example, right? So it's all about the dose. In terms of mechanisms, how could too much alcohol potentially be harmful? There's many potential mechanisms, but one of them seems to be this dehydration cycle, where you drink a lot. And Dr. Jicha may speak to this as well. We've noted in our autopsy consensus conferences sometimes that you can actually see changes to the brain in someone with a history of a very heavy alcohol use. So our best guess is that a little alcohol, mild to moderate, is not probably going to affect you negatively, but certainly the possibility is there.
Thank you so much. Moving on then, and please, other panelists, if you want to add to any of the discussion, I ask you to jump in. What type of testing is used to diagnose Alzheimer's? And so, Dr. Jicha, that might be a good one for you to address, but also Marie, if you have anything to add, that would be really helpful.
I'm happy to address that, because often there is this black box of, oh my goodness, I think I'm having memory problems. What in the world is going to happen to me in there? Is this going to be a frightening experience? And I'm happy to say that many folks that I see for initial concern about memory may be completely normal, or may actually have a medical condition that's responsible for that memory decline. It could be inappropriate medicines that they're on that we can simply switch. It may be your thyroid being a little out of whack, or a vitamin deficiency. So in clinic, we have a very straightforward way. We bring people in, we talk with them about their concerns, about their problems. We move into a physical examination to make sure we're not finding anything medically that could be wrong with someone, a neurologic examination to make sure that there aren't other things. And these are all very simple, like listening to the heart with the stethoscope and hitting people on the knees with the rubber hammer. And we all know that from visiting with the doctor. Maybe some brief memory tests. And if we see a memory problem there, that may be enough for us to move forward with. We can always send people for more intensive memory and thinking tests, the kind of tests that make us all feel like we have Alzheimer's disease because they're so hard to complete. There's a purpose behind that. It's really to see where people are on the scale of normal performance. And then sometimes we do have to go further. And Dr. Levy alluded to this with some of the research that sometimes there are brain imaging studies that need to be done. Sometimes we have to balance what those costs are versus what the information we're going to get. But sometimes we will send folks for PET scans. Typically, a CT scan or an MRI is very helpful for us to look for vascular disease and the like. And using all of that information, it's kind of like being a detective. We're kind of pulling that all together to come up with, is there a problem or is there not a problem? And if there is a problem, what is that problem so that we can best design a way to treat and a way to hopefully slow that progression. If it's a medical cause, perhaps reverse it. So demystifying that black box of, oh my God, Dr. Jicha's going to put me in a nursing home. That's the last thing in the world I or any other clinician wants to do. What we want to do is help you not have a memory problem and or to slow a memory problem if it exists so that it doesn't wind you up in a nursing home or debilitated or unable to enjoy your quality of life in the future.
Thank you. Marie, anything to add?
I would just add, and usually Dr. Jicha is very strong in saying this as well, we have to partner with families as we're doing all of that evaluation of a person who may be having some signs of memory impairment. We know from lots of years' experience in seeing folks that they may not recognize that they're having an impairment. And so we need that input from folks who are spending length of time with them, and so that we can get a timeline that gives us a better feel for, did this start suddenly? Has it been going on for some time? Have they always been a little off and now they're more so? Those pieces. So we need to, I think a crucial part of all of this care of folks with memory impairment is partnering with the families, working side by side with them through all of it.
Thanks, Marie.
Donna, if I could just add, I had a chuckle. I had a chuckle when Greg said we all feel somewhat impaired when we take those detailed memory tests. It's absolutely true. We have 1,500 people that come in every other year for memory tests and spinal tests and 8 hours of testing. They universally would rather have a spinal tap than have the memory test.
And having taken those memory tests myself, I felt very stupid when I was taking them and thinking, oh my God, I'm failing this. So yeah, I agree, but that's like Dr. Jicha and everybody else says, they're valuable. And I'm assuming Dr. Levy at Emory, you guys use very much the same process that was described. And we do have guests from all over the country, from Florida and California. So wherever you go, it will be a very similar process that gets worked through. Okay, so then moving on, and I might actually ask Dr. Van Eldik to contribute to this as well. So Dr. Levy spoke about inflammation in his presentation. And so one of the questions is, we say inflammation a lot in medicine, but what does it mean on a cellular level, if anything? Is it damaging the cell wall or the receptors on cells, or am I off? And so, no, I would say no, you are not off. But maybe Dr. Van Eldik, who is a world-renowned expert on inflammation in the brain, can tell us a bit more about that.
Sure. So inflammation is a word that is not well defined. because there are a lot of aspects of inflammation. And as you saw from Dr. Levey's presentation, there are a lot of things that change when we talk about inflammation in the brain. Some of those changes that occur are beneficial inflammatory changes. Some of them are detrimental inflammatory changes. And so we're trying to understand which things can we control selectively? Can we block the bad inflammation, but keep the good inflammation? And also the problem with inflammation and targeting inflammation is that it occurs at different times during the disease progression. And so there are some inflammatory changes that may occur at a time where you want to keep those changes. And then other inflammatory changes occur either earlier or later in the disease, and you'd want to block those. So we know that there are certain responses that are bad, and we would like to modulate those, and that's kind of the approach that my lab is doing. But we also would love to kind of beef up the good inflammation and hit the bad inflammation. So we certainly don't want to have a very broad suppressor of all inflammation, like steroids or medicines like that. That would not be good. So we're developing selective ways to hit the bad inflammation. And the field it's in a state of progress. There are a lot of different strategies being used right now to target inflammation.
I'll just add that I'm really hopeful when you look at how the cancer field is getting to use the same mechanisms. It's one of the most exciting new areas for treatments for cancer, right? And just learning how to control the immune system and inflammation.
Thank you. Hopefully that demystified it a little bit. It is an evolving field, and we're learning more, it feels like, by the day about inflammation in the brain. So a question for Dr. Levy and Dr. Jicha. Should a person currently being treated for high blood pressure ask their cardiologist about switching to an angiotensin receptor blocker like candesartan? So Dr. Levy, you want to take that first?
It's a great question, and the most important thing is that you work with your doctor on an individual basis. In my view, certain medicines are better for certain people, so I wouldn't take where we are in research right now to be prescriptive about what people should more broadly do. But it's a great question. I will say, when I went on, I was newly diagnosed with hypertension a couple of years ago. It led me to want to be on candesartan, which I'm on.
Yeah. So always very interesting, and I'll just echo what Dr. Levy said. There are some medical conditions that are contraindicate the use of an angiotensin receptor blocker. So sometimes degrees of kidney impairment may make the doctor shy away from that. Sometimes a little bit of kidney impairment actually makes them move more towards candesartan. But when we get this early suggestive medicine, the early suggestive results from these trials that this medicine or that medicine may be useful, I think it's worthy of a discussion with your doctor, because if we're sitting in a state where either or of multiple medicines could be beneficial for you, you may want to say, well, the wealth of evidence may suggest that this isn't may lead to positive benefits. And while I don't know for sure if it's 6 of one and half dozen of the other, it's worth of that discussion and to see what your primary care doc says about that decision. Ultimately, they're going to make the decision that is best for your blood pressure and or your other medical condition. And over time, with increased research, if we can be more definitive about some of these early leads that are moving us forward, we may be able to be more definitive in recommendations.
Thank you so much for that. Question for Dr. Abner. We heard a little bit about asymptomatic people who may have, plaques and tangles in their brain, but don't really have symptoms yet. Can you talk about how frequently that really occurs and just what that might mean to dementia research?
Yeah, so again, it's one of those things that's not completely one way or the other, right? So there's a continuum of how much amyloid we have in the brain and where it is, and the same thing for tau. We do see, though, that there are probably far more people than you would expect who, when they pass away and we look at their brain, they have a decent amount of Alzheimer pathology, and they were still doing fine, right? And so what does that mean? It means a few things. One is that there does appear to be some sort of natural mechanism that we have that we can withstand some of that pathology, right? So and that level is probably different in different people, considering your overall health and your age and your lifestyle factors, all of those things. But the thing I want to be clear about is when we see a lot of Alzheimer pathology, we talk about the amount of tau pathology in the brain and its distribution in terms of something called Braak stages, and those go from 0 to 6. And so if we have a brain that has a Braak stage 6, that's the most severe neurofibrillary tangle tau burden that we can have, and we do not see people withstand that, right? So there's some level that some people, in fact, many people can withstand, and there's a certain threshold beyond which we can't withstand that. And so as Dr. Levy was saying, it would be fantastic, right? So if we have this disease process that's ongoing, but we don't necessarily have symptoms, if we can push those symptoms down the road, that's better quality of life, that's better time with your family, that's all really important. So the fact that these people exist and that this is pretty common is another reason for hope, right? That this is not the end of the story. And Dr. Jicha is going to comment as well.
Yeah, I had to throw my hand up into the ring because, and I think that Dr. Levy alluded to this as well, is that we don't know if all people developing amyloid are truly going to go on to develop Alzheimer's or not. Maybe they just passed away before they developed Alzheimer's. Maybe they could withstand that indefinitely. But we do know that the early buildup of that pathology is part of the disease process. And so a lot of work with these medicines is not just finding new medicines, but taking some of the medicines we have that can reduce amyloid, keep it below that threshold that Dr. Abner was talking about, and actually identify these folks. And we don't have to wait until autopsy. Again, Dr. Levy talked to us about PET scans and spinal fluid. So we are actively engaged in a very large multi-center trial called the AHEAD study, where we're bringing folks in and looking to see if they have elevated amyloid in their brain. And if they do, offering the opportunity to potentially receive a medicine to reduce those levels. And this is a question that we have to answer in the field. We know that once the disease has progressed too far, there's not a lot we can do about it, but if we could keep it from that tipping point, would that be helpful? This is critical for us to understand. We also have other studies. If we find that somebody is having early memory problems and has no amyloid or tau in the brain, maybe that is late. The disease that Dr. Nelson here has popularized, and we have active clinical trials as well to try to stop those disease processes. So again, if you're interested in engaging in fighting this fight with us, there are ways that you can help, and we need you to join with us. We can't do this alone. We need everybody to stand up and say, no more, no more. Let's do this for ourselves. Let's do this for our children. This is important.
Thank you so much. So just very quickly, Dr. Jicha, could you comment on the relationship between late and Alzheimer's, seeing as that was mentioned by you just now?
So a very interesting topic, and and for years, and everybody understands this historically, throughout our lives, years ago we used to just say, well, they're just senile. And then we all jumped on, well, that's hardening of the arteries, right? And everybody had hardening of the arteries. And then in the 1980s, everybody had Alzheimer's disease. Well, we've seen through the research advances that Dr. Levy showed us that we have become much more sophisticated. If you went to the doctor and the doctor said, well, you have a heart problem, the first thing out of your mouth would be, what kind of a heart problem do I have? Because we need to fix this. And that is exactly the way that we approach your brain. So Alzheimer's disease clinically is a pattern of memory loss and decline in daily function, but there are mimics, other diseases biologically that look like Alzheimer's disease, and late is one of them. And late can come as a pure form, frequently seen in people in their 80s and 90s, but more often it's actually seen in conjunction with Alzheimer's disease. So we'll have a little bit of Alzheimer's, we'll have a little bit of late, we may have some other pathologies thrown in the mix as well. And Dr. Abner has really driven the field in this area with what she describes as QMP, quadruple misfolded proteins. We may actually need multiple medicines to treat an individual person in the future. We may need medicines for late and for Alzheimer's disease. We may need, within Alzheimer's disease, anti-inflammatories, anti-amyloids, anti-taus. That's where the field's going very much. And I think Dr. Levy alluded to this before, maybe it was Dr. Abner, the field of cancer in terms of how do we treat cancer? It's not one medicine or one chemotherapeutic combination for an individual patient. It is looking at that individual patient and saying, no, which mix of treatments might we need? And so I think that's the relationship between late and AD. They can occur distinctly, but much more often they're occurring together. And we really need to embrace that concept if we're going to move the field forward and really begin to help people who are developing early memory problems.
I'll just add, Greg, that was a great description. We're really excited to be collaborating with you and Dr. Nelson, with our same brain proteomics and spinal fluid and blood. We would love to be able to find some other ways to detect late in living people. So we're eager to get going on that. So it's great to work with you guys.
And we're getting up and running, Allan, and we appreciate your partnership in that effort.
All right, thank you so much to both of you. Now, I want to do the next few questions as rapid fire if we can, so that we can get through as many before the time runs out. This would be for Dr. Levy and Jicha. We have questions about supplements, and we have a specific question about B vitamins. So could both of you comment very quickly about what we know about supplements and B vitamins?
Go for it, Greg. I'll let you take it.
So I always say, if you're going to start a supplement, talk to your doctor about it. The supplement itself might be completely harmless, but it might interfere with your other medicines. I also see a large number of people on heavy doses of B vitamins. There are some B vitamins, water soluble. You can't overdose because you're just going to urinate them out. But other B vitamins like B6 that are lipid soluble can build the toxic levels in the body. Please consider your nutritional supplements like a drug and report them to your doctor so that we can make sure there are not adverse effects of that. You don't want to do more harm while you're trying to do good and stay healthy.
I'll just add very quickly, we know that too low levels of some of these vitamins like B12 can be a problem and a cause memory loss. And as Greg said, we know that too high levels of some vitamins can be toxic. So great advice from Dr. Jicha.
That's great. Thank you. Is it possible there will be a vaccine for Alzheimer's disease? And this has been in the media a little bit because of a new study on an intranasal vaccine that started, I think, this week.
I think it's certainly possible. People are still working on that. The new study that you alluded to, as I understand, it's not necessarily a vaccine against amyloid, unlike where most people are focusing. It's a vaccine to more generally enhance our immune system's ability to fight a variety of pathologies. So.
Yeah, that's great. I think possible, but not in the very near future.
And we'll talk about that, Donna, in the next section.
I thought you might, so that's good. This is a very important question, I think, as we move towards early diagnosis with our biomarkers. How might diagnosis affect future health insurance benefits? And I'm not sure who wants to take that one, but if anyone has a comment on that, it'd be great to hear.
I think Marie may want to field that a little bit, working so closely with families that we do have to take these issues into consideration. When are we going to put a diagnosis in the chart? How do families need to prepare, and patients need to prepare to have that diagnosis in their chart?
Yeah, I think that pretty well covers it. That needs to be a very it's something we have to look at very carefully and individually with folks, and where they are now in terms of their coverage and that sort of thing, and then to move forward and see.
Yeah.
That's going to be a real. It is a challenge now, and it is going to become, I think, more of a challenge.
I agree.
I just jump in with a follow-up question for Marie. So what would you say to someone who's concerned, right? If their loved one has potential memory problem, but they're concerned about this insurance issue, how do they move forward?
I encourage families to help that person and the family themselves to look very carefully at what their coverage and their status is now, because there are times when we need to make changes in what our insurance coverage is. And then I also encourage families to look at not just insurance, but all of that person's resources, and to make sure that everything's in place that they can have in place to offer them protection currently, in terms of being able to use those resources wisely, and then moving into the future. Again, it can be very complicated.
Yeah, thank you. So last question, which I hope is a very quick answer from all of you. How would someone who is potentially interested in getting into some kind of research study, even at the younger age of 40 and 50, how do they go about doing that? And this is UK, but also, as we know, we have people around the country on with us right now. So please let us know.
I'll chime in just quickly on that. First off, if you're in our area, call the center. If you're not in our area and you can't find anything, call our center. We will try to help connect you with what's available in your area. There are also large national programs that have been developed. Alzheimer's Association has Trial Match. The CTSA initiative has Research Match. We have an APT web study going on where you can be tested cognitively if you're over the age of, I think, 50. So there are many different ways. And as we move towards earlier and earlier diagnosis and treatment, and that may just be diagnosis of future risk, not diagnosis of a disease state, the opportunities for people younger and younger to participate are growing exponentially. Dr. Levy, or Levy, you're very active on the national scene. Other thoughts on that?
Yeah, no, I agree. I was going to put in a plug for one of our studies also, which is online. for especially those younger people that Donna mentioned, 40s and 50s. It's called the Emory Healthy Aging Study, and it's just going online to help give us information to help us understand risk factors and in the future ways to connect. So we have about 30,000 plus people that are online, come in annually, just sign up and do about 20 minutes of questionnaires and try to provide a lot of information about brain health promoting activities. And more of those are popping up. So certainly not the only one.
That's great. And Dr. Abner did put the website for that into the chat. So please, if you're interested in learning more, check that out. There are questions still left in the Q&A. I left them in there. Hopefully our panelists will be able to go in and type some answers to the ones we didn't get to. But in the meantime, thank you for everyone's really great questions. And Dr. Van Eldik, I'll hand over to you.
Great. That was a great panel discussion. Thank you, guys. And now for the piece de resistance, as they say, we're going to have a discussion from Dr. Jicha and Dr. Abner to talk about Educanumab, which Dr. Levy introduced to us in his talk. So I'm not sure who's going first, but go ahead and get started on that.
Let me see if I can get my slides up and running. And Dr. Abner, if it's okay with you, I'll run the slides. You can just tell me when you want to advance yours. Well, fantastic. So Dr. Levy really set the stage today and presented a bit of this information. We've all heard a lot on the news about the FDA approval, conditional accelerated approval, we'll get into that in greater detail, of one of the first potential disease-modifying agents for Alzheimer's disease. I'm delighted to co-present with Dr. Abner on that. I'm just going to set up a little bit of background for us. Again, which Dr. Levy has already set up. You know, we've been looking at medicines to potentially cure, or at least treat and or slow Alzheimer's disease for years. And we've been focusing at this very late stage of disease when we know that we may need to be focusing much earlier, certainly for the amyloid therapies, that may be critical. And we're not sitting still. You know, many people don't realize that, you know, it was well over 2 decades ago that the first Alzheimer vaccine was introduced and actually given to people. And unfortunately, that vaccine was far too strong. And while most vaccines are safe, every now and then we do hit one, and we identify that very early in the experimental stage where nope, this can't move forward. And that was the case at the time in 1999. The Alzheimer's disease vaccine trial was halted because it caused some inflammation, swelling in the brain, an inflammatory process. So sometimes a little inflammation can be good, but inflammation out of control, very, very dangerous. Over the next decade, people had worked on, well, how can we potentially do this safer? How can we have better control? And that led to the era of passive immunization. And that is where Educanumab really comes into play. It's true, there's not just one type of amyloid in the brain, but amyloid can be seen in many different forms. And maybe it's the form that makes the difference in terms of what we need to target. And again, as one of our prior questions addressed, are vaccinations on the way? So the first thing I'd like to just lay out there for everybody is, you know, there is more than just one type of vaccination. And we broadly group these into 2. Active vaccination, that's, you know, kind of like our COVID vaccine. You get a shot in the arm and you're protected against COVID. And passive immunization. Passive immunization is an infusion of antibodies where your own immune system does not have to make the antibodies to fight off the disease. Instead, we can provide those antibodies. We can slow it down, we can speed it up, we can increase your dose. If you're having trouble or side effects, we can decrease your dose. Gives us a lot of control. And so that really, given safety issues, is where the field has relied on that tight control to exactly try to understand how we can best modify the disease. But we're not stopping with our goals for active vaccination. There have been over 14 new vaccines developed for AD that target amyloid, and other vaccines, as we've heard, like the nasal vaccine, targeting broader mechanisms that are involved in Alzheimer's disease. The newer vaccines all appear safe for the most part. We're not seeing any major safety signals, but as of yet, we've not seen high enough immune responses from the aging population to really move these forward. We're continuing to work on that over time. And that is the hope someday, that we'll have a vaccine. When you turn 50, maybe, we'll all go in, and instead of just getting the shingles vaccine, we'll get the AD vaccine, maybe the late vaccine as well. So I do want to just talk briefly, this is my commercial segment on COVID vaccination. The COVID vaccines have been proven safe, and COVID has taken a tremendous toll on us. And in the lower left, you'll see data from our own group here, who we polled in the COVID crisis. How problematic is COVID to you? And over 80% of people worried about this. And I think that should be 100%. I'm worried about it. Almost 90% feeling isolated. Almost 100% feeling that this has been incredibly disruptive to my life. How am I going to go to the gym and get that exercise that I need to get? And 80% of people feeling completely out of control. The vaccine can get us back in control. And so on the right, this is a study done in California of healthcare workers who work directly with people who are COVID positive, as I do in the hospital as well. And if you are fully vaccinated, the likelihood that you are going to catch COVID is extremely low. If you're partially vaccinated, that still helps. That would be one of the 2 shots, or maybe 2, but not a booster. But if you're fully vaccinated, this is going to be beneficial. If you're not vaccinated at all, you're going to be at high risk. So please, please, please talk to your doctor. There are virtually no reasons why someone should not be vaccinated. There are very, very few. The vast majority of us can be vaccinated. Millions have been vaccinated, millions and millions, hundreds of millions worldwide. This is safe. Let's do it for each other. Let's do it for those that we love, especially as we approach the holiday season. So I'm going to chime in again on COVID and move back to passive immunization. We all know President Trump caught COVID, and they whisked him off to the military hospital. And at the time, we didn't have a vaccine, and it would have been too late for him anyway. Instead, they used a medicine called remdesivir. This is antibodies given in an infusion or an injection that helped Trump beat COVID in just a couple days. This same strategy is what we're doing currently with passive immunization. Again, thinking there are several different antibodies out there. We're going to focus on Educanumab, but there are several like it. And we have to recognize that these medicines are not all the same. They catch amyloid in different ways. Some may target the plaques, some may target the monomers, different forms of amyloid. And the one thing that we can see is for most of these medicines, the stronger they are, the more potential for side effects. We need to find the right place where safety is less of a concern and efficacy, the ability to potentially slow Alzheimer's, is at its best. Now, I like to, kind of describe this as how you might catch a rattlesnake. And I say that because it was one of my favorite activities when I was a kid. We used to go out rattlesnake hunting. And that may sound dangerous. My parents would just roll over if they knew that that was what we were doing out in the woods. But there are different ways to grab a rattlesnake. You can grab it in the middle of its body. Well, there's a bigger target there. It's easier to catch, but that snake may just turn around and bite you. You can try to grab it by the tail. I don't want to get anywhere near its head, but again, it might whip around and bite you. The best way to grab it is right behind the neck so that you don't get bit. And so we're trying to think about this kind of a strategy with these different antibodies. What's the best way to potentially catch amyloid? How can we do this moving forward? That's where the field has been developing, and that's really what's led us to 2021, a time of celebration for all intents and purposes, even though this is not the cure we're looking for, the first FDA approval of a disease-modifying agent. But we really need to understand this information. This may not be for everyone. And so Dr. Abner is going to walk us through what that data actually shows.
Thank you, Dr. Jicha. So I want to start with just a quick overview of why we randomize people in randomized clinical trials. What is the purpose of that? What does it do for us? So let's imagine that we have two groups of people who are enrolled in a clinical trial, and maybe we have two treatments, one called A and one called B. So the idea of randomization is that it doesn't actually matter which group gets treatment A and which group gets treatment B. So we may plan in the study to give group A treatment A, and we may plan to give group B treatment B, but there could be a mix-up, and instead we give everybody in group A the treatment B, and we give everybody in group B the treatment A. But we don't expect this to really impact the results of our clinical trial assessing the treatment effects. So randomization makes it so that it doesn't matter which group of people get the treatment. And so this means that we can conclude that any differences we see between those groups of people are due to the treatment and not due to characteristics of the people themselves. Next slide, please. Right, so we can make strong causal inferences about treatments and outcomes. So that's why clinical trials are so valuable. Next, please. So I'm going to talk about, as you've seen already a couple of times, two clinical trials that Biogen, the drug company that developed Aduhelm or Educanumab, ran to assess the efficacy or the effectiveness, right? How well does the treatment work on clinical endpoints? And those were called the Engage and Emerge trial. And the design of those two trials was very, very similar, purposely so. And so Biogen presented results from those two trials in 2019 at the American Academy of Neurology meeting. And so I'm just going to repurpose some of those slides. If you're interested in seeing the full slide deck, the URL will be available. So Emerge and Engage, 18 months long, right? Randomized, we know why. Double blind, meaning the participants didn't know who got what treatment and the investigators didn't know who got what treatment. And we have a large group of people, a large number of study sites. We have data from 20 countries. That's all very exciting. And the people that were enrolled in the study are people with what we think of as very mild cognitive impairment or early Alzheimer's disease. The population that you can see there on the screen, the two pieces of information there, MMSE 24 to 30, those are considered normal to mildly impaired scores. The same for the CDR global score of 0.05. And they're also requiring that there's a confirmation that the participants in the trial do have amyloid accumulated in the brain. Another important thing to note is that it wasn't just drug versus placebo here. They were really looking at two versions of the possible dose for Aduhelm, what they'll call low and high. So they had a placebo group who didn't get any Educanumab or Aduhelm. They had a group that got a low dose, and they had a group that got a high dose. And so, as it says here, their primary endpoints, which is very important in a clinical trial, you have to specify those up front and you can't change. Otherwise, it's difficult to know, again, whether it was actually the treatment that caused what you saw or whether you have just tortured the data until it tells you what you want to hear. So we are looking for changes in the CDR sum of boxes, which is related to that global CDR score. And we're looking for these other cognitive endpoints, the MMSE and the ADAS. Next slide, please. And so I just am showing this slide to show that when you get this treatment, this shows that you have to what they call titrate up. So we can't just start people out on the maximum dose because you can get some negative effects. So we start them at a low dose and sort of increase them. And then ultimately the dose that was approved by the FDA is this high dose that I have circled here. Next slide, please. I want to turn to the ability of the participants to complete the study. And so these data are always reported in clinical trials, and it's important to think about them. So we can see here in the two trials that at the dose that was approved, we had about 550 people randomized to that dose. And we can look at this data and see that about 24 to 25% of the people who were randomized to that dose had to stop taking it. They could not complete the trial because of a variety of reasons. And so those reasons are listed here, and of concern to us are adverse events that cause people to discontinue treatment. And certainly there can be other reasons that are not related to the treatment in particular. The study site, for example, was closed in some cases. In some cases, the participants were just too busy. It was too difficult to fit the study into their lives. So it's not all about safety, but it does concern us again that about 25% of the groups discontinued the treatment, and also that at the end of the day, we've only got about 50% of the people who started this study completing it. Now that's not really different across the treatment groups, so that's not such a concern about safety, but it does give us a little bit of a concern about how well people might be able to actually use this medication in real life for the rest of their lives when 285 people, right, were all out of 547, for example, in Emerge, were able to complete the study. So that's somewhat concerning. Next slide, please. And again, thinking about safety, so we always report this information, very important. And an AE is an adverse event, and those can be expected or unexpected, and those are really any change, any new condition, any worsening of a condition that you had at the beginning of the study. And so it's not surprising to see that number of adverse events, 505 participants, for example, out of 547 in the Emerge high dose group had an adverse event at some time during the study. Not particularly surprising, so that's not really a concern. What we are concerned about are participants who permanently discontinued the treatment because of an adverse event. And so we're looking at about 10% again of the groups that were assigned to that high dose could not take it because it caused problems for them. Next slide, please. And so what are the problems that the medicine caused? And the ones that we're really concerned about here, and Dr. Jicha will talk more about, is this ARIA that you see on here that I have highlighted. So amyloid accumulates not only in plaques that we saw pictures in Dr. Levey's presentation, it also accumulates in the blood vessel walls. And the anti-amyloid therapies don't discriminate, at least at this point, between amyloid plaques and amyloid that's in blood vessel walls. And so when we get amyloid being pulled out of the blood vessel walls, sometimes that causes swelling and bleeding in the brain. And we see that that was very common, right, at the high dose. We have 35%, right, of each of those high dose groups experiencing this brain inflammation. And we have an additional 20% experiencing brain bleeds as a result of getting this treatment. So we are concerned about that. This can be a very mild or asymptomatic experience for patients. It could also be lethal. So it's very important that people who are taking this medication be monitored very closely to make sure that this is not happening. Next slide, please. I also want to point out that we use this randomization to say, well, if we treat this group of people versus this group of people, and we see a difference in their outcomes, then we know that it's due to the treatment. But can we assume that the same results would be observed if we included people who were not like the people that were in the trial? So who was not included in the trials? So nobody that had any medical or neurological condition that might cause cognitive impairment could be included. Nobody who had had a stroke or a TIA or fainted in the last year could be included. No one with psychiatric illness. I'm not sure exactly how they operationalized unstable, but that includes a whole lot of people, right? Depression, anxiety, heart conditions excluded, impaired renal or liver function excluded, and that's very common in older age. No one with HIV infection or any other type of infectious illness, right, within the past 30 days. Makes sense. We wouldn't want people with a history of brain bleeds in the study. It might be dangerous for them, so they were not included. People who couldn't go in a scanner to have a picture of their brain taken were not included. People who are heavy users of alcohol or other substances were not included. And again, that's a large population. And people taking blood thinners, right, could not be a part of the trial. Next slide, please. Also very important for us to think about the mix, right? The racial and ethnic mix of the people in the study. So that's a big problem for these studies. We saw the map, the worldwide, we have all of these study sites and thousands of participants. But at the end of the day, there were only 6 Black people in the entire 2 studies, Engage and Emerge, who were randomized to that high dose. So we have very little information about people who are not white, right? And then in terms of Asian Americans, we have very little information as well, because although almost 10% of the study participants were reporting Asian race or ethnicity, they're actually from Asia, right? Taiwan, South Korea, and Japan. So our information on a lot of very large populations is very limited. And so that's a concern. Next slide, please. We've seen these slides a couple of times. I just want to go back to them again. So this was the primary outcome. This was the thing that the clinical treatment effect was pinned on this, right? We had to see an effect here. I've added in these red horizontal lines, which kind of show you the difference. And so this treatment effect in the Emerge trial, so versus placebo, which is the gray line, it's the one at the top, and the purple line is a little less than half a point on the CDR sum of boxes, which is a very small difference, probably not clinically important. We wouldn't expect differences in quality of life, functional status, or really cognition at that level of difference. And then in the Engage trial, you can clearly see that it's hardly there at all. And again, we have just a subsample of people who started the trial. So not very compelling evidence that this medication will help cognition. Next slide, please. On the other hand, there's very compelling evidence that the medication reduces the amount of amyloid in the brain, and that's what we're seeing here. So the difference between the gray lines, which are the placebo groups, and the purple lines are very clearly different. And again, I want to note the number of study participants who contributed data to this analysis is actually quite small at the end of the day. And that is not necessarily because of dropout. They only planned to do this amyloid analysis in a sub-study, right? So in about little less than a third of the people in each of the study groups, they were going to do the amyloid sub-study. So we have information showing us very clearly that amyloid is reduced in about a third of the participants who were supposed to be in this study. Next slide, please. I don't expect you to read this. It's just a screenshot from clinicaltrials.gov. This is somewhere that you can go and find out what the investigators said that they were going to do in a study, which is what they should do at the end of the day. And their primary and secondary outcome measures I'm just showing are all cognitive. So nothing about the amyloid here for what we're trying to do in this trial. So these trials are designed to show a clinical benefit, right, on cognition. Next slide, please. But they didn't, right? So next slide, please. So FDA approves Aduhelm on the basis of its ability to remove amyloid from the brain, not at all its ability to improve cognition. But in my view, there's really no evidence at all that it improves or slows cognitive decline. Next slide, please. So this is under the accelerated approval mechanism, where we can have the FDA say, we don't have clinical evidence that this treatment is effective, but what we have is evidence that a biomarker or a surrogate endpoint is effective. And so the FDA says it's reasonable to think that because Aduhelm does remove amyloid, that it will at some point in the future clinically benefit patients. And so this accelerated approval mechanism gives Biogen 9 years to submit evidence that Aduhelm does work in a clinical sense. Next slide, please. So what's the big deal then? Why are we upset, or why, people are saying we're not prescribing this, very limited use. So there was an expert panel you may have heard that recommended against approval. As I've mentioned, the trials were not designed to seek accelerated approval. They were for clinical endpoints. There's some evidence that there were inappropriate sort of relationships and partnerships between certain FDA officials and people from Biogen, the company that makes Aduhelm. And of course, Aduhelm is wildly expensive. The drug itself is $56,000 per year. And so our concern is that Aduhelm won't help people, and it can actually harm some people. And if a lot of people take it, it can potentially harm a lot of people. Next slide, please. We're done. Thank you.
Very good. Very good. So I'm not going to reiterate much of what Dr. Abner said for the sake of time. You've seen these slides that she has shown. We now, from her careful description, understand that it engage and emerge data. But one of the things that's come up in the field is that similar agents, similar antibodies, and here I show two. In the top, I show Lecanemab or BAN2401, again on the left, lowering amyloid in the brain and showing a potential clinical benefit, still questionable, but a potential benefit, slowing of progression of disease. And in the bottom panel, Donanemab from Eli Lilly, again, removing amyloid from the brain on the left very effectively and influencing outcomes on the right. This is beginning to build circumstantial evidence that as a class, these medicines may actually have some benefit. And so people are beginning to look at the pooled data. Both of these agents have already been filed with the FDA for approval. We're waiting to hear what the FDA says about these. My best guess is they will be approved conditionally in an accelerated fashion, just like Educanumab. So we already know accelerated really means we need to do something, and we need to do it fast, and we all want acceleration, but we do need to understand. We know these medicines will remove amyloid from the brain, but we do need additional work. We need additional work to prove their benefit, not just over 18 months, but maybe the benefits, are going to magnify over time. We don't know that, but we may learn that in a post-marketing phase. Again, Aduhelm, not for everyone, so it requires a careful discussion. And with any doctor that's going to prescribe this, really, I've been approached by a lot of folks with much more severe disease. This is not a medicine for them, unfortunately. It's not a medicine for Lewy body disease. And you may need spinal fluid and or a PET scan to show that you have amyloid in your brain. We don't want to give you a medicine to remove amyloid if you don't have any in your brain. And everybody's going to need an MRI scan so that we can monitor for that ARIA that Dr. Abner talked about. And these are just pictures of the brain showing this, with Educanumab, now brand named Aduhelm, the rate of which one could experience either swelling of the brain, we can see in white over here on the left, or these little black dots are microhemorrhages, small bleeds in the brain, can be as high as 41%. We need to watch. And again, that's why we use passive immunization. If we start to see these effects and there are symptoms from them or they appear severe, we stop the medicine, hold it for a little while, or reduce the dose. And then when the brain recovers, we sometimes can restart it, sometimes we can't restart it. But very important, safety is number one every step of the way. And again, those coverage concerns, $56,000 a year. Several centers have already declared we're not going to give this at all. Medicare, though, is reviewing this currently for a coverage analysis that they expect to be done by the spring of 2022. Typically, drugs undergoing this process historically always get approved. What would that mean? Well, Medicare Part B coverage is typically only 80% of a medication's cost. So we're still left with 20% of $56,000, potentially not very cheap for folks. And we really need to fix that. So lots of issues here. Again, I think the take-home points are we have a lot more work to do, but we are seeing potentially some early promise in changing the disease. You need to get educated. You need to get informed. You need to find a center of expertise and have these discussions if you're interested in this medicine. And again, like the first man on the moon, this is the first step. But it may be a giant leap for humankind. And let's see. There are many, many, many more medicines on the way, but we've broken through that threshold to actually begin to bring these Alzheimer medicines we've been working on for decades further in the treatment of people in the community. And that's important. And what's next? The possibilities are just amazing to me. There are well over 100 potential new treatments, including many that could be possible cures under investigation in experimental trials now. We're at the leading edge of this research. We have these opportunities. We need us all to pull together right here, right now. Let's get involved. Let's find out if any of these are right for you. Always keeping your safety and well-being first and foremost. So I really appreciate your time today. Again, Educanumab is not the cure we've been looking for, but is it the first step in that direction? I have to be excited at this point. We will be hopefully bringing the opportunity for Educanumab to the University of Kentucky. It's under review currently, but again, it's only going to be able to be administered by experts that can watch your safety and well-being. And it's not going to be available for everyone. You're going to have to meet very specific criteria, and we'll do our best to work with you to figure out if this is right for you, or if we should approach your memory problems in a different way.
All right, thank you very much. And thank you to everybody who participated today. Thanks especially to Allan for spending the entire symposium here with us and participating. And I just wanted to remind the audience, when you sign off, you're going to get a little automatic survey that pops up. Please fill it out. It's very helpful to us. It'll only take a few minutes. And I just want to thank everybody who participated today. I think this was a fantastic symposium. And hopefully fingers crossed, we'll be able to do this in person next year. So thank you, everybody. Enjoy the rest of your day.